Hello, EV enthusiasts. Welcome to the WebEVTalk series, the 3rd February 2022. So it's fantastic to meet you again for this first session of WebEVTalk. I hope everyone is in good health. So now it has been almost 2 years since the start of the COVID pandemic, and we are now moving toward living with COVID as expected, with the virus continuously mutated. So that brings us into our work and our love affair with the field of extracellular vesicles, or EV, as we like it or not. If we think about clinical relevance for our future studies, we must consider the correlation or connection between virus and EVs. So today we have with us Carolina Balbi, who is one of the first EV scientists to publish research on EVs and COVID infection. So Carolina is a young scientist, and she managed to pivot from her research from a myocardial infarction field to look into the role of EVs in COVID infection. I think her work and experience that she's sharing today, looking into this topic, will help all of us to understand further and figure out the scientific questions for the field of EVs moving forward in COVID normal situation. So before we start, I actually have a poll here. So I'm just going to try to start the poll. Let me see. This can be sort of answered anonymously. So if you can quickly answer that. It's just for a survey purpose for myself. So can you answer that, please? Thank you. There's actually about 30% of us here that doesn't work in EV field, and half of the participants actually work in COVID-19. So that's interesting. So that means that this topic today is quite popular, and it will be really one of the focus moving forward. So I'm just gonna stop the poll now. So at the end of the meeting, there will also be a survey, and I would really appreciate of getting help from you by answering it, as it will give me some prompt to how to redirect WebEVTalk moving forward. Because as we now in a traveling and face-to-face meeting is becoming more and more common, I have asked myself repeatedly whether WebEVTalk as virtual platform is still loved and needed by EV enthusiasts. So if you like WebEVTalk, please leave a comment on the survey, or maybe for those who watch it through YouTube, you can also leave comments at the YouTube and like the video today. So thanks everyone, and without delay, I would like to welcome Carolina with her talk entitled "The Role of EVs in COVID-19 Infection." So, go ahead, please.
Perfect. So good morning or good evening, depends on the part of the world you are. Everyone, I'm really happy to be here and to present my work on extracellular vesicle and COVID. I'm really happy to be the first of the 2022 series of the WebEVTalk. Thanks really again, Carolina, for the invitation. And today I'm going to talk I'm going to give you an overview of what is present in the literature about the COVID-19 virus infection and the extracellular vesicles. So just a little bit about my past research and what brings me to study extracellular vesicles and the relation between vesicles and COVID. I started my research activity in 2015 with my PhD at University of Genoa, where I studied the role of the secretome of human amniotic fluid stem cells in a model of myocardial infarction. So treating myocardial infarction with the secretome of these cells. With secretome, we refer to soluble proteins secreted by the cells, but also to extracellular vesicles. So during my PhD period, I started to new extracellular vesicles. I became interested in extracellular vesicles, and after my PhD, I moved to my postdoctoral lab, where I'm still working in Cardiocentro Ticino at Laboratory for Translational Research of the Ente Ospedaliero Cantonale in Switzerland. Here I focused my research still in extracellular vesicles and their role in the treatment of the heart, but vesicles from another source of cells, from cardiac mesenchymal progenitor cells. As all the extracellular vesicle enthusiastic person, during the COVID-19 outbreak, I became interested in the role of extracellular vesicles in COVID infection. We knew that coronavirus is able to infect the cells to the binding of this spike protein to the ACE2 receptor. This binding leads to damage of the cells, and in particular to endothelial damage, because endothelial cells are the first cells infected by the coronavirus. And this endothelial damage leads to inflammation and a cytokine storm. There is a huge relationship between inflammation and extracellular vesicles. You can find in literature a lot of paper with the role of vesicles during the inflammation. And as a sample, I can show you here two papers. In the first one, the author treated the cells with glia cells with LPS, so they induced inflammation in the cells and then characterized the vesicles secreted by these cells. And as you can see here, there is a change in the cargo of the vesicles in inflamed cells, a change with increase in inos, NLRP3, and procaspase 1 cargo in present in the inflammatory milieu, and also a change in the dimension of the vesicles. In the second work that was done in another lab from the Laboratory for Translational Research at Ente Ospedaliero Cantonale, where I work, in a myocardial infarction model in vivo, they found that there is a huge increase in extracellular vesicle production in the circulation of the animals after myocardial infarction, but not only an increase in number of vesicles, also an increase in a change in the cargo of the vesicles with, again, pro-inflammatory protein enriched in these secreted vesicles. So it's reasonable to think that also in COVID-19 infection, there is an increase in secreted vesicles from cells from the immune system, but we can also find in the circulation and also extracellular vesicle produced by infected cells, for example, vesicle produced by endothelial damaged cells. We have the possibility to perform, we are an hospital, so we had the possibility to perform the analysis from the serum, in the serum of a patient from COVID-19 positive patient. And for this reason, we decided to characterize the vesicles of this patient. And as you can see here, we took the serum sample from COVID-19 and COVID-19 positive and negative patient. So all the patients were symptomatic and underwent the nasopharyngeal swab test, and then we divided after the result of the test from a positive and negative patient. The serum was precleaned with low speed centrifugation by cell debris and large vesicles, and after the precleaning we perform a labeling of the serum with fluorescent beads labeled with 37 different antibodies, and then we perform flow cytometry analysis. First data that we found is this one. We found an increase in the number of the vesicle from COVID positive compared to the COVID negative patient. So confirming that the huge inflammation leads to an increase in the vesicle secretion by different type of cells. Among the different markers expressed in the surface of the vesicles, we found really interesting the different expression in CD142 in a COVID positive patient. CD142 is better known as tissue factor, and we know that tissue factor is one of the components of the coagulation process. And looking at the literature, but also looking at the study reports, is known that thrombotic complications are frequent in COVID-19 and patient and contribute really significantly to mortality and morbidity for the patients. So we focus on tissue factor, and first of all, we confirm that it's really overexpressed in COVID positive derived extracellular vesicles. As you can see here, was confirmed by Western blot after the isolation of the vesicles. And we also found that it's not only overexpressed, but it's also overactivated. So there is also an increase in the activation of tissue factor on these vesicles. The activation also is correlated with the expression, of course. As mentioned before, in the circulation, we can find the vesicles derived from different types of cells. And thanks to the clustering analysis of the surface marker of the vesicles that we analyzed by FACS, we found that during in the COVID positive patient, there is an increase in vesicles derived from the endothelium. So really suggesting that the damage of the endothelium is important and leads to a secretion of a huge amount of vesicles. So this was the first work that we performed. It was done by me and by Jacopo and Sara, and the lab was the lab of Prof Barile in Cardiocentro Ticino. This data of the expression of tissue factor was also confirmed by different other papers. In this one, they found that not only there is a correlation, there is an increase in tissue factor expression in extracellular vesicle from derived from patient COVID-19 positive patient, but also that there is a correlation between the severity of the disease and the expression of tissue factor, as you can see here. And furthermore, that this expression of the tissue factor is really related to COVID, not to every type of infection. This is something that we also found because we had a COVID negative patient, but there were symptomatic patients, so patient that has an influenza, a different type of influenza. And again, in this other paper, they confirmed by FACS analysis the expression. They found the overexpression of tissue factor in the microvesicles, circulating microvesicles of COVID-positive patient. As mentioned before, these vesicles have possibly also a role in the disease, so they are secreted and they can cause where they can increase the damage, they can propagate the disease, and that was studied by this by Dillon lab, and they isolate the vesicles from COVID positive patient at different stage of the disease, so from asymptomatic to severe patient, and they found that both large vesicles and small vesicles were able to induce endothelial apoptosis when these vesicles were used to treat endothelial cells in vitro. So they can propagate the damage and they can increase the endothelial damage when circulating in the patient. They also perform a proteomic analysis on these vesicles, and again, what is interesting is that they confirm overexpression of tissue factor in extracellular vesicles derived from COVID-19 patient. And also they confirm that the activity of tissue factor is significantly correlate with the length of the hospitalization and the disease severity of both the large and the small extracellular vesicles. So there is really a huge correlation between the expression of tissue factor and the severity of the disease. Now I want to show you this other paper that is really interesting. Still, there is a characterization of the extracellular vesicles derived from the blood of the COVID-19 positive patient. And here the authors found that this extracellular vesicle carries some fragment of the spike protein, the famous spike protein of the SARS-CoV-2. Interesting, what they found was that there is an increase in spike protein in vesicles derived from mild patient compared to the severe one. So there is higher enriched the S protein in these vesicles compared to the one derived from the severe patient. And they also perform again proteomic analysis of the vesicles from the mild and the severe patient. And what they found was that the cargo of the extracellular vesicle from mild patient is a cargo of protein that have the possibility to proper activate the immune system. So they are important to good activation of the immune system, while protein cargo of the severe extracellular vesicles derived patient are protein that leads to a chronic inflammation. So they are important to, are a cause of increased damage of in the patient. It's important to know that not only the protein are an important cargo for the extracellular vesicles, so we all know that also nucleic acid, but also lipids are important. And in this nice paper from Nature Metabolism, the author decided to analyze the lipid, they performed lipid analysis on the extracellular vesicle from circulating, derived from circulation of patients with COVID-19. And they again analyzed the vesicles from pre-symptomatic patient, infected patient in the hyperinflammatory phase, infected patient in the resolution phase, and recovery patient. And as you can see here, there is a huge dysregulation of the lipids in the vesicles derived from the hyperinflammatory phase patients. And this dysregulation leads to a different expression of membrane protein, as example PS1. And this different expression in membrane protein also leads to different activation, pathway activation in target cells. So they again isolate the vesicles, treated endothelial cells with these different vesicles. And for example, they found that there is a complete opposite activation in Notch pathway if they treat the endothelial cells with vesicles derived from hyperinflammatory phase or vesicles derived from a patient, recovered patient. So not only the protein that is are important for the role of these vesicles, but also the lipids composition of the membrane. Last paper that I want to show you regarding the circulating extracellular vesicles is this one, which is really, really interesting. Here the author decided again to isolate the vesicles from patients with different stage of the disease, from healthy people and also from patient asymptomatic, mild and severe patient. But they decided to isolate the vesicle 3 months later, after the 3 months after the recovery. So when the patients were already fine and there is no trace anymore of the disease. And as you can see here, there is a signature in the vesicles after 3 months. There is an enrichment of some proteins. As example, TGF-beta1 is enriched in the extracellular vesicle from COVID-19 former positive patient. And this is not related to the severity of the disease. It's upregulated in all the patients from asymptomatic to severe one. For other protein, the story is different. They are still upregulated or downregulated compared to the healthy people, but there is also a difference between the different stage of the disease. That is really important information, because that suggests that is not the disease is not done when the patient is recovered, but there is still something going on. But talking about extracellular vesicles, there are, it's important to underline that extracellular vesicles and COVID are not only studied looking at the role of the vesicles in the disease, but there is also a huge amount of study that try to use extracellular vesicles to treat COVID-19 infection. And the treatment is can be done with different strategies, for example, using modified extracellular vesicles or using mesenchymal stem cell derived extracellular vesicles. As example, one of the idea that was suggested in the scientific community was to use spike protein expressing extracellular vesicles. This these extracellular vesicles can be important to decrease the virus infection because they can competitively bind the ACE2 receptor in the target cells, decreasing the virus infection. Spike protein expressing extracellular vesicle was already generated from Bussolati lab, as you can see here in this super nice picture, with in green the spike protein, in red CD63, and in blue CD9. They generate spike protein expressing extracellular vesicle to study the interaction between the host cells and the spike protein. But for example, this other paper, they try to see if really there is the possibility to compete with the ACE2 receptor between the virus and the spike protein extracellular vesicles. So they again generate vesicles expressing spike protein on the surface, and they found that really this vesicle can fuse to the target cells only through ACE2 receptor, thanks to ACE2 receptor. As you can see here, without ACE2 expression on the target cells, there is no fusion of the protein of the extracellular vesicles, but with the ACE2 expression, there is a fusion between the vesicles and the target cells, and this fusion is abolished when we use an antibody against neutralizing antibody against spike protein. So this could be a strategy to counteract the virus infection. Another strategy is to express ACE2 on the membrane of the vesicles. This will lead to a viral binding and decrease, again, the viral infection. Also, this was done in this paper where the authors generate the ACE2 expressing extracellular vesicle. And as you can see here, using target cells plus the virus, without extracellular vesicle, there is an infection of about 15% of the cells. With the co-treatment with EV expressing ACE2, there is a huge decrease of the viral entrance in the target cell. So also this could be a really nice proposed strategy. But I cannot stop my presentation without talking about the mesenchymal stem cell derived extracellular vesicles, because I started with mesenchymal stem cells. It's well known that mesenchymal stem cells derived extracellular vesicle has an important role in immunomodulation. For example, I'm going to show you two examples, but really there are thousands in the literature. Here from a paper from former colleague of mine in the University of Genoa, they isolate the extracellular vesicle from the adipose tissue, mesenchymal stem cells, and they found that these extracellular vesicles are important to induce the polarization of the macrophage to the pro-resolving M2. So they have an important role in anti-inflammatory effect. And here, this is another paper from my PhD, again with the human amniotic fluid stem cells derived extracellular vesicles. We found that also these vesicles have immunomodulatory properties. So as I said before, these are only two examples, but it's really well known and well established the role of mesenchymal derived extracellular vesicles with anti-inflammatory potential. And for this reason, if you go to clinicaltrials.gov, you can find till now 10 registered clinical trials that propose to use extracellular vesicle from mesenchymal stem cell to treat COVID-19 positive patient. Among these clinical trials, 3 are already completed, and I'm going to show you the results from one of these, in which the they show that after 5 days of infusion of extracellular vesicles derived from bone marrow mesenchymal stem cells, there is a significant decrease of some important parameters, such as a decrease in CRP, ferritin, D-dimer, and ANC in the treated patient, and also an increase in the number of the circulating lymphocytes from day 0 to day 5 after the injection, after the treatment with the systematic treatment with the extracellular vesicles. So really there are important data and that suggests that this could be a good strategy for the treatment of the patient. But I have to be honest, there are not only the mesenchymal stem cells, there are also mesenchymal progenitor cells, as example, the cardiac mesenchymal progenitor cells, the one that we use it in the lab. These cells, we derive these cells from the human heart, that we obtain a piece of tissue from the surgery, from patients that are undergoing to heart surgery, of course, and we derive these cells, and we already used these cells in different disease models, as I said before, in cardiotoxicity, myocardial infarction, and this vesicle showed to have really an important therapeutic potential. But what I want to underline now is that during this study, we found that also the immunomodulatory potential of these extracellular vesicles is really important. There is, for example, a decrease in the CD68 infiltration in the heart after myocardial infarction when we treat the heart with extracellular vesicles derived from cardiac mesenchymal progenitor cells. And importantly, this effect is also higher than the one from the bone marrow derived bone marrow mesenchymal derived extracellular vesicle. And in this paper, every comparison was done with the extracellular vesicles derived from cardiac mesenchymal progenitor cells from one patient, and from the same patient also the bone marrow derived mesenchymal stem cells. So it's not related to the cell, to the patient, is related really to the what is produced by the cell. So from is related to the vesicles, let's say. So because we are the Cellular and Molecular Cardiology department, and the heart is of course our favorite organ, I have to say. If we look at coronavirus, we cannot ignore that coronavirus have tremendous effect on the heart. There is a huge increase in the myocardial infarction risk, heart failure, arrhythmia, and there are evidence that the virus can really, as you can see here by this microscopy analysis, can really infect the cardiomyocytes, so the cells of the heart. So I can say that we can suggest to use cardiac mesenchymal derived progenitor cells, extracellular vesicles, to treat COVID-19 positive patient, and then especially with the one with heart complications. So we can hit two birds with one stone, I have to say, and we can try to ameliorate the situation, the inflammatory situation, and protect the heart. What is important is that we know that systemic injection of these extracellular vesicles is able to reach the target organ, and in particular these vesicles are able to target the heart, so they really can be functional for the treatment of a COVID-19 positive patient with heart complication. So to conclude, I think I already talked quite a lot. The role of extracellular vesicle in COVID-19 virus infection is really, really interesting. There is a huge topic around the role of the vesicles in the disease, so what they can do, how they can increase the severity of the disease, and but there is also a huge topic regarding the role of the vesicle to treat the COVID-19 positive patient. So there is really something that we can take into account, and we can start to investigate more. So with this slide, I would like to thank the Laboratory for Translational Research at Ente Ospedaliero Cantonale, my lab, and in particular the Cellular and Molecular Cardiology department, the one in which I work, and the Cardiovascular Diagnostic department from Professor Barile. And of course, all of you for the attention. Stop sharing.
Thank you so much, Carolina. Wonderful presentation, and thanks for summarizing it for us. It's really a huge compilation of what is happening right now with the COVID-19 studies and EV's role in the coronavirus infection. So I'm opening the QA session right now. If you want to just raise your hand, I can also prompt you to start asking questions. And probably start with my own question. So you're using the you're using the CD9 antibody to capture and assess the EVs in the COVID-19 infected patients. Have you tried the different antibodies and whether there's interesting differences or results on that?
So what we used was the cocktail of CD9, CD63, and CD881 together. So we never try to separate them. So we don't know if there is some association between, because there is also a paper that shows the role of CD9 in the COVID infection, not in the COVID, in the SARS-1, let's say. They found that CD9 is important for the virus entrance. So it could be interesting to see if there is a relation, but we never try to separate them. We only used the combination of the three together. So we don't have data.
Yeah, so it looks like if it really does carry the tissue factor, that sounded like it's accentuate the symptoms for the COVID-19 patients. So what do you think about using EVs as COVID vaccine then? Would that be sort of like dangerous, or do you have some sort of thought about it?
You mean vesicles derived from COVID patient injected in patient as a vaccine? I really don't know, because they show to carry, for example, some part of a spike protein, but it's not really clear if it's inside or if it's in the surface. So it's not something already quite clear. I don't think is a good strategy. Maybe it's good to use extracellular. It could be an idea to use extracellular vesicles as a vaccine, but modifying extracellular vesicles, for example, to express on the surface spike protein in order to produce antibody, but not the one derived from the patient. I think it's not a good strategy.
Yeah, so I guess it's sort of like the using non-autologous source like the MSC, like you're trying to do at the moment.
Exactly. They are already was already proven in different diseases, but they have no problem in the injection between patients. So they are completely safe, let's say.
I have one question here from Anne Grosso. Please, can you turn on your video and also unmute yourself? Hello.
Hi, great talk, thank you. Yeah, I was just curious to see if there was any difference between the different COVID variants when it comes to COVID patient positive with COVID, sorry.
Yeah, this is really, really interesting. We don't have data regarding this because the first paper that we did, the first work that we did was from the first wave of COVID. So I think the most severe one, let's say, but it's going to be, I think it's really interesting to compare, for example, the Omicron variant, what is produced by the bodies of the vesicles that we can isolate from Omicron variant infected patient compared to the wild type one. is something really, really interesting. We don't have data now, but I think we should move to start to collect the blood sample from this patient. And it's interesting also to see if the vaccinated people have different production of extracellular vesicles. So it's a good suggestion, and I think that we should move forward in this direction. Thank you.
So with your study looking into the targeting the heart, can you tell us a little bit details about it? What is the source of the EVs and other things?
Yeah, so the lab in which I'm working have a huge, quite a long story with the vesicles derived from the cardiac mesenchymal progenitor cells. They are cells derived from the heart tissue. These cells have all the mesenchymal markers, so they are CD100, 05 positive, CD73 positive, CD90 positive. And we already characterize a lot these vesicles and we used this vesicle to treat myocardial infarction or other disease, for example, drug cardiotoxicity. We knew quite a lot of this vesicle because we already performed proteomic analysis. So we know that they, for example, are able to have on their surface the protein periostin protein, and periostin protein was already shown to be important to bind integrins on the cardiomyocytes and is related with the cardiomyocyte regeneration. That is something really important after the damage. So yeah, this is the main topic of the lab, and as I showed, we know that these vesicles are able to target the heart. So if they are injected systematically injected in the animal model, we can find these vesicles on the heart. So I think could be really a good strategy. Furthermore, I didn't mention in the presentation, but at Cardiocentro Ticino, the hospital in which I'm working, they already developed a GMP method for the production of this vesicle. So there is, we already tried this vesicle in a pig model. Is quite forward the study of this vesicle, so it could be really easy to move to a pathology, to another pathology, and try to treat a COVID positive patient with these vesicles.
Yeah.
So yeah.
I think one of my sort of burning question is like, when we looking into EV profile in COVID infected patients, is there any data that suggests that the viral cargo can be detected in the EVs as well?
So we tried a lot. We also tried to perform digital PCR on the isolated extracellular vesicles to see if we can find some viral RNA in the vesicles, but we cannot find anything, I have to be honest. We also tried, but in the beginning, we also tried with the antibody against spike protein, but yeah, I have to be honest, the first antibody were not so good, now are really better. And in the end, we moved to the tissue factor analysis because we found this interesting correlation. But in now there is some paper that show that there is part of spike protein carried by the extracellular vesicles. It's not really clear if it's inside, outside, but or they talk about fragments, so it's not really the full protein. But yeah, it seems to be, for sure there is not DNA, not RNA. That is something that we really tested a lot, and is not present RNA in the vesicles. So yeah.
Yeah, it's very cool. Yeah. Do you have any suggestions for people who wants to start looking into COVID infected patient in their diseases, whatever their disease that they're working in? Do you have any sort of like a top tips, I guess, to share with us?
Yeah, first things is that is not really a tips for the related to COVID, but it's a tips, I think, related to everyone that works with the patient. I think one of the most important things is to have real important guidelines to the to the handling of the blood sample, for example, or from the other sample, because every small differences can lead to differences in your extracellular vesicle preparation. And also some guidelines in which you want to, what you want to see, and so the timing of the sample collection and everything. This is a tips for every type of study, but still is also with COVID, it's really important because the different, as I showed, the different stage of the disease leads to different vesicles production. And I think that there is still a huge of work that we can do with for the study of vesicles in the COVID-19 positive patient. And also now we are really, we know really a lot more of the disease, and as suggested by the first question, looking at the different variants is something interesting. There is a lot of work that we can do, and I think it's really, really interesting. And I can only suggest to do what you like and let's say working on your passion.
Definitely. Other questions from the participants? Otherwise, perhaps we close the session soon. No? Okay. All right. With that, I'd like to conclude and also thank Carolina for sharing your result today and also giving us an educational talk about the role of EVs in COVID-19 infection. So for this year, the WebEVTalk is not going to run every week unless there are people who come to me and tell me that I really want to give a talk. But I wanted to sort of really focus on particular papers and particular topics that are getting really hot right now and getting very interesting. So the next talk is not next week, but on the 22nd February in the USA or 23rd February in the Asia Pacific, which is Masaharu Somiya from Japan. So he's going to talk about developmental of experimental tools to investigate EV cargo transfer and intracellular communications. So again, thanks for joining us today. And as a reminder, after the session over, there will be a survey coming up. If you can answer that, that will really helpful for me to organize the next sessions and how the program going to be moving forward. And with that, I end this session. And perhaps if anyone really keen to be involved in WebEVTalk, you can stay in this channel for a while, just and then can ask me any questions, and we can possibly get you some experience in science communications or whatever other organization that WebEVTalk does. So thank you so much. So please take care. Thank you, Carolina.